All right, so first I'd like to thank Dr. Rose for the invitation to talk to you today. And so my lab has been interested in, can everybody see the pointer? So my lab has been interested in understanding the underlying neurobiology of nicotine addiction for over a decade. And like many groups, we began focus on the traditional mesolimbic reward pathway. And as we all know, in this pathway, there are dopaminergic neurons in the VTA that project to the nucleus accumbens. And all drugs of abuse, nicotine included, acutely causes an increase in dopamine release in the accumbens. And this is associated with the rewarding or reinforcing effects of the drug. In my lab and a host of others have spent a great deal of effort trying to understand the nicotinic receptor subunit composition or subtype that's most responsible for nicotine's rewarding effects in this pathway, with the idea that if you can identify a specific subtype of nicotinic receptor and you may be able to design a specific antagonist or partial agonist in an effort to block the reinforcing effects of nicotine as a smoking cessation strategy. But more recently, my lab and a host of others have begun focusing on a much more understudied circuit. This is the medial habenula interpeduncular axis. And within this circuit, there's the medial habenula, which is an epithalamic brain region, and it projects almost exclusively to the interpeduncular nucleus of the midbrain, which is very closely, neuroanatomically close to the ventral tegmental area. It's just medial and ventral. And within the medial habenula, there are a cluster of neurons that synthesize and release acetylcholine, and they also co-release glutamate. And this is the major excitatory input to the interpeduncular nucleus, whereas the IPN itself is an inhibitory nucleus. More than 80% of the neurons in the interpeduncular nucleus are in fact GABAergic. And so what's emerging from multiple groups is that this circuit is absolutely critical for nicotine dependence. And this was first highlighted by Paul Kenny's group. And so what he found was that if you knock out the alpha-5 nicotinic receptor subunit, or knock down this subunit, which is highly enriched in this habenula interpeduncular axis, you get rodents that will self-administer nicotine at doses much higher than a wild-type animal, indicating that this circuit is critical for nicotine intake and regulating nicotine intake. Whereas my lab has found that if you, that this circuit is very important for nicotine withdrawal. So if you take a mouse and you give it chronic nicotine, so it's dependent on nicotine, and you take that nicotine away, what you get is hyperactivation of the interpeduncular nucleus. And this triggers physical withdrawal symptoms, but more importantly, the affective withdrawal symptoms, including increased anxiety. And so we've even shown that if you silence the medial habenula input into the interpeduncular nucleus during nicotine withdrawal, you can alleviate or block anxiety. But what's interesting about this circuit is, although we know now that it's critical for nicotine dependence-associated behaviors, its role in the context of normal behavior is still virtually unknown. But what we found recently is that this medial habenula IPN circuit, which is emerging as an aversion circuit, and the mesolimbic pathway, which we know is the reward pathway, may be directly connected. And so we found that there are neurons within the VTA that are dopaminergic that actually project into the IPN, suggesting that these two circuits are coupled. And so when thinking about the role of the medial habenula IPN pathway in the context of normal behavior, we may be able to get some insight based on what's known about the ventral tegmental area in behavior. And although we associate the VTA with drugs of abuse, one of its best known roles is in the response to novelty and novel stimuli. So when presented with a novel stimulus, this activates the mesolimbic pathway, causing an increase in dopamine release in the nucleus accumbens. And this motivates an animal to explore the novel stimuli. And this is a critical critical for survival, and then it motivates exploration of new stimuli which may present an animal with opportunities for new food sources, for example, a chance at mating, a novel social encounter. But over time, with repeated presentation of the novel stimulus, this dopamine response is decreased as a novel stimulus becomes familiar. And the mechanism for that is unknown, and also the circuits involved in familiarity are not very well described. And we know that this is also important for addiction because novelty seeking is associated with nicotine sensitivity, for example, heavier smoking and lower quit rates. And we also know in animal models that higher novelty preference correlates with addiction phenotypes. And so based on our connection between the VT and IPN, we wondered if the interpeduncular nucleus was also involved in the response to novelty. And so this is the question. And to address this using mouse models, we focused on what is a very robust novel stimulus in mice, which is social interaction. And so to look at this, we use a 3-chamber social interaction test where we have a test mouse in the middle here. And it's in a chamber where there's 2 cylinders. And in one of the cylinders, we put a novel social stimulus, which is a same-sex juvenile mouse. And normally what you find is that on the first day of this test, the test mouse will explore the novel stimuli more than the control, which is just an empty cylinder. But on the second day, what just happened here? As the novel stimulus becomes familiar, there's less interest and less exploration of the social stimulus. And then on the third day, there's even less interest of the test mouse because now what was once a novel stimulus has become quite familiar. But you can get rebound exploration if you put a new novel stimulus in the cylinder. And summarizing what I just said here, on the first day you get this increased exploratory activity. By day 2, that activity goes down because the novel stimulus becomes familiar. On the third day, you get even less interaction with the novel stimulus. And then if you put a new novel stimulus in the cylinder, there's a rebound in investigation. And we can look at this as a preference ratio. And the important thing here is, for you to know, is that as the preference ratio goes to 0, that essentially just means no preference. So the test animal will explore the cylinders equally. So the first question that we ask here is simply, we know that the VTA is activated by novel stimuli. Is the IPN activated by novel stimuli? And to look at that, what we did was we just exposed an animal to a novel stimulus, a social stimulus, and then we isolated the brain, sliced and stained the IPN, looking for CFOS as a marker of neuronal activation. And what we were surprised to find was that after a novel social stimulus, we did not see any difference between CFOS expression in the IPN compared to the control. However, if we presented the social stimulus multiple times so it became familiar, we saw an increase in CFOS in the IPN, suggesting that the IPN is not activated by novelty, it's actually activated by familiarity. And in fact, the more familiar a stimulus was, the more activation of the IPN we saw. And importantly, as I mentioned, this is a GABAergic area, and the majority of neurons where CFOS was expressed were GABAergic. And this led us to our hypothesis here, which is that familiarity activates the IPN, this inhibitory nucleus, and perhaps that reduces exploratory activity towards a novel stimuli. And so to test that hypothesis, we turned to optogenetics. And the first question that we wanted to ask was, we wanted to know if we inhibit this activation of the IPN, does that affect novelty preference? And so we used halorhodopsin, which is a light-activated chloride pump, so that we could selectively silence and hyperpolarize GABAergic neurons. And we injected that in a viral AAV, viral-mediated gene delivery into the interpeduncular nucleus of GAD2 mice. So these are mice that express Cre only in GABAergic neurons. And this is a Cre-dependent halorhodopsin, so that they will only the GABAergic neurons will express halorhodopsin upon injection. And when we do that, we see nice halorhodopsin expression in the interpeduncular nucleus. And this is actually from a mouse after behavior. So you could see the fiber tract targeting the IPN, and halorhodopsin is expressed only in GABAergic neurons. And more importantly, in slices, if you patch clamp onto the GABAergic neurons and you apply yellow light, you can shut them off. And so you can see here that we can silence action potentials in slice. And so to look at novelty preference, we did this test where on day 1, we take our test mouse and we expose it to a novel social stimulus. On day 2, we expose it to the same social stimulus, so it becomes familiar. And then on day 3, we expose the mouse to the same social stimulus, so now it's very familiar. But in the other cylinder, we put in a novel mouse. And typically when you do that, what you see is that a normal test mouse will prefer to explore the novel animal, as shown here in the heat maps, compared to the familiar animal. But if we turn off the GABAergic neurons in the IPN, the test mouse now explores the familiar mouse and novel mouse relatively equally. And so that's summarized here. So if we look here, this is the time spent investigating the familiar social stimuli. And what you could see is that if you turn off the GABAergic neurons in the IPN, there's an increase in exploration of the familiar mouse as if it were novel. But this has no effect on investigation of the actual novel animal, presumably because novelty silences GABAergic neurons in the IPN, and you don't get any further effect by turning them off with halorhodopsin. And as a consequence, this reduces the preference ratio closer to 0, so that the mouse can no longer discriminate between what is novel and familiar. And importantly, this doesn't affect total investigation time, so we're not just affecting locomotor activity, for example. What does this mean? We know that novelty has a rewarding component, a motivating component. And so what we did was we wanted to test if perhaps the IPN plays a role in that. And so we took a conditioned place preference, which is our last speaker told us is usually used for drugs of abuse to determine if a drug of abuse is rewarding or not. And we modified this conditioned place preference during training. So instead of a drug of abuse, we paired one chamber with a novel social stimuli and the other with a familiar social stimuli. And then on the test day, asked the mouse to decide which chamber does it prefer. Does it prefer the chamber where it encountered a novel stimulus or where it encountered a familiar stimulus? And normally what happens is a mouse will tend to spend more time in the chamber where it encountered the novel animal during training, whereas it does not prefer the familiar chamber. Familiarity is not aversive, it's just neutral, so there's no effect. But if we turn off the GABAergic neurons in the IPN during the training phase when the animal's paired with with a familiar animal, when GABAergic neurons are activated, if we turn these off, what happens is now the test mouse will spend more time in the familiar paired chamber where we turn off the GABAergic neurons. And so this suggests that the IPN GABAergic neuron activity is necessary for the decreased motivation or reward that's associated with familiar exploration. So what happens if we do the opposite? What happens if we activate these IPN GABAergic neurons during novelty preference? So to do this, we use optogenetics again, but now we use channel rhodopsin, which will allow us to, with light, control the activity of GABAergic neurons in the IPN and make them fire. And what we find is in the novelty preference test, if you activate GABAergic neurons in the IPN, this has no effect on the interaction with the familiar mouse but it significantly reduces interaction with the novel mouse. So now the test mouse is interacting with the novel mouse as if it were familiar. And this reduces the preference ratio essentially to 0, which is now the test mouse doesn't explore the familiar mouse that much, but it also doesn't explore the novel mouse that much because it's recognizing it as familiar. And what about in the place preference test? So now in this experiment, we're activating GABAergic neurons in the IPN during the encounters with the novel mouse during training. And so once again, in a control animal, normally an animal will spend more time in the chamber where it encountered the novel stimuli. But if you activate GABAergic neurons during training with the novel mouse, with the novel stimuli, you essentially prevent this motivation response, and you prevent the preference. And we think that what's happening here is that these GABAergic neurons are reducing the rewarding effects of a novel stimuli. And so based on this, we found that familiarity actually activates GABAergic neurons in the IPN, and this reduces novelty-induced exploration, and this is because it demotivates the animal to explore what once was a novel stimuli. And in a series of experiments that I don't have time to talk about, we found that this familiarity signaling is mediated through the medial habenula, so it activates these excitatory inputs into the IPN to activate the GABAergic neurons. So now what about the ventral tegmental area? So as I mentioned previously, we found evidence that there may be a connection between the dopamine neurons in the VTA and the IPN, and we know that these dopaminergic neurons are activated by a novel stimuli. So we wanted to know, is the VTA really functionally connected to the IPN, and what effect does this have on behavior? And so to test that, we used optogenetics again, and this time we expressed channel rhodopsin in dopaminergic neurons in the VTA. So we injected DAT-Cre mouse, so this is a mouse that expresses Cre recombinase only in dopaminergic neurons. And when we do that and we look at YFP expression from the YFP tag connected to channel rhodopsin, and we image the IPN, we see that there are terminals that are dopaminergic that innervate the interpeduncular nucleus. And if we stimulate them with light in slices, we see that in GABAergic neurons, there's a modulation of neuronal activity when we stimulate these VTA inputs. And we find that these are almost always blocked with the D1 receptor antagonist, suggesting that these IPN neurons express the D1 receptor. We rarely see D2 responses. How does this affect behavior? Well, to test that, we looked at a mouse where we're stimulating the dopaminergic terminals with light that are in the IPN. And during novelty preference, what we find is this increases the interaction of this test mouse with the familiar social stimulus. So in other words, it's interacting with the familiar stimulus as if it were novel when we activate these dopaminergic inputs. And this has no effect on the interaction with the true novel stimulus. And that again reduces the preference ratio essentially to 0 because the animal's exploring both the familiar and novel mouse equally. And is this mediated by the D1 receptor? To test that, we did this experiment where we implant a fiber optic cannula to deliver light to the IPN, but we implant a dual drug infusion fiber optic cannula so that we can infuse a drug selectively into the interpeduncular nucleus before we stimulate the dopaminergic terminals there. And when we do that, if we infuse vehicle before we stimulate terminals, we see a high interest towards the familiar social stimulus because we're stimulating dopaminergic terminals in the IPN. But if we pre-infuse a D1 receptor antagonist into the IPN, we can block this effect. And essentially, this restores our preference ratio back to the mouse actually preferring to explore the true novel stimuli compared to the familiar stimuli. And so based on that, we know that familiarity through the medial habenula activates GABAergic neurons in the IPN, and this reduces novelty-induced exploration and the motivating properties of novelty. Whereas novel stimuli itself, we know activates dopaminergic neurons in the VTA, and some of these dopaminergic neurons project to the IPN, and we think that this essentially keeps the IPN quiet to promote novel-induced exploration. And so we think this is going to be very important in the context of nicotine dependence, because we think that chronic nicotine activates this pathway and essentially keeps the IPN quiet. But when you take away nicotine after chronic treatment, this leads to a rebound excitation of GABAergic neurons in the IPN, which essentially causes behavioral avoidance and withdrawal symptoms. And so right now what we're testing is we're testing the effect of chronic nicotine on these new dopaminergic inputs to the IPN that we've identified to see if they're dysregulated by chronic nicotine treatment. And so based on that, I think the people who did the work, so almost this whole novelty project was driven by Susanna Molas, who's a postdoc in the lab, currently on the job market, with help by a longtime research assistant, Professor Rubing Zhao-Sheh, and all the physiology was done by a very talented graduate student in the lab, Steve DeGroot. These are my funding sources down here, and I'm happy to take any questions. Thanks a lot.
Thanks for a wonderful set of studies. And Jed has a question.
Thanks. It was a really elegant set of experiments, and in that the effect of either activating or suppressing IPN activity, either GABAergic cells or dopaminergic, have such different effects, then it really becomes critical what a given dose of nicotine does. And you said you think nicotine chronically will activate the GABAergic, I take it GABAergic cells?
So I think that the chronic nicotine at low concentrations, the rewarding doses of nicotine will activate dopaminergic neurons, preferably in the mesolimbic reward pathway, and that will keep the IPN quiet. So the only concentrations of nicotine that activate the habenula IPN pathway are extremely high. And so normally I think a rewarding dose of nicotine would not activate this circuit.
Okay, that was my question, is what a cigarette smoking like dose, kind of a transient, bolus kind of dose would do, but
I think
you
it would
don't see activation of habenular neurons with anything but super high doses. Or the other thing is, could they be desensitizing the habenular neurons from the actions of acetylcholine?
They could be. They could be. We haven't tested that directly, but the only, and so right now what we're doing is, so most of what we've done has been using CFOS as a marker of neuronal activation, and so that really limits what you can get out of your data. But what we're doing now is we're doing in vivo calcium imaging experiments using calcium sensors, and so we're measuring the response to nicotine in vivo in GABAergic neurons of the IPN, using rewarding doses of nicotine. And so we'll see. I mean, we'll know soon, I think.
Thank you. That was very interesting and definitely very elegant way of going about to answer these interesting questions. I was wondering if you have any ideas or comments about other forms of learning that are not social dependent.
So we know that the medial habenula IPN pathway is involved in associative learning in fear conditioning, for example. And so there's definitely a learning component, and obviously there's a learning component in an animal having to decide if something's novel or familiar. But we think that the IPN is downstream of hippocampus and cortical areas. And so we think that at least the IPN is more involved in the motivating behaviors associated with novel stimulus or familiar stimulus. But there's definitely a learning component, and we're in the process of mapping that now.
Andrew,
Yeah,
first off, I'd like to thank you. Elegant piece of work, but
Thanks.
at least you provided a partial answer to a question that has bugged me ever since 1978. Most of you weren't born then. But one of the things that we discovered very early on is the interpeduncular nucleus has the highest concentration of nicotine binding of any region in the brain. And for the longest period of time, at least this old idiot had no idea what the IPN did in general, and certainly how it interacted with nicotine. I do have one question, however, and it's something that I think you ought to think about, and the previous speaker needs to think about. And this is a 76-year-old man who does not remember the author of the relevant paper. But the relevant issue is putting a mouse or a rat in a box multiple times turns out to be an anxiolytic stimulus. And I worry about the interpretation of some of the results in the last two talks in terms of that issue. So for example, in your data where you were seeing increased activity in the box, blah, 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 same one actually for the same previous talk, is are we seeing the anxiolytic properties of nicotine emerging in a test where we keep testing them and it's more and more anxiogenic and we've given nicotine, and so this increase in activity is observed. And it's not saying that your data are wrong. Your data are absolutely right. But I think you need to do a little bit of, is Al nuts? Does this paper never been published? Or if he's not nuts, can I think about my data in a little bit different way? And the reason why I think this is important with respect to the smoking issue is people have talked about for a long, long time that I smoke to reduce anxiety. So it's a different form of reinforcement. It's not dopamine released in the nucleus accumbens, but it sure as hell seems to be important for why people smoke. Wonderful work, Andrew. Wonderful work.
thank you. So I'll address your question. And so in all the experiments that I showed you, especially in the conditioned place preference, there was no nicotine on board. So this was simply activating or deactivating GABAergic neurons in the IPN. And you're absolutely right that anxiety can influence this behavior. And so the IPN is involved in anxiety. It's a very small part of the IPN called the interpeduncular intermediate, which is activated during nicotine withdrawal, and that actually triggers anxiety. But in response, in the context of the experiments that I just showed you, we did measure anxiety-like behaviors in the open field, in the elevated plus maze, and on marble burring, while we activated or inhibited GABAergic neurons, but I didn't have time to show you. And the answer is that whether we activate or inhibit these neurons, it had no effect on any of the anxiety measures that we looked at.
I had a question.
Yeah.
So in terms of the circuitry in the brainstem, the cholinergic innervation of the VTA coming from the dorsolateral tegmental nucleus and the peduncular pontine nucleus, I assume these are the same sources for the IPN, right?
No, so the IPN gets almost all of its cholinergic innervation from the medial habenula, but the GABAergic neurons that I showed you in the IPN, we have mapped to the lateral dorsal tegmental area. So the IPN actually provides inhibitory input to the lateral dorsal tegmental area.
I see.
And we're in the process of functionally characterizing those inputs.
So all of these nicotinic receptors in the IPN, so the cholinergic innervation is coming from cholinergic cell bodies in the habenula?
Yeah.
Yeah. Huh.
And it's frequency dependent, so those neurons also release glutamate. But in order to get them to release acetylcholine, they need to be stimulated very strongly.
I see.
Yeah.
Okay, well, thank you. So now we're going to have a break, morning break for 15 minutes. We'll reconvene at 10:45. Okay, thank you.